Hey, how are we doing everyone? Welcome to the Bearded Medic. In this video we will go through the action of the various T-cell subtypes after they've been activated as part of the adaptive response, and we'll be looking at all the cytokines and transcription factors involved. So we had our dendritic cell presenting our foreign pathogen, and it was recognized as foreign by a T-cell with a specific T-cell receptor, so we had clonal selection of that T-cell. Co-stimulation occurred, and this T-cell was encouraged to survive and replicate into a certain subtype. Now this subtype depends on the cytokines released onto the naive cell in the differentiation process. T-cell responses can be split into 5 types. These types are the T helper 1, the T helper 2, the T helper 17, the T follicular helper, and the T regulatory. Each of these have their own cytokines and transcription factors involved. Now this sort of immunology has little context. It can seem like a bunch of random numbers and letters, but to make it easier, organize things as flowcharts. So for each of these subtypes, they have a polarizing cytokine that upregulates a transcription factor that lead to other cytokines being released and having actions on certain cells. It's also important to note that these polarizing cytokines can be released by a range of cells, and we'll explore this further as we go through the video. So let's begin with the T helper 1 response. The T helper 1 response responds to microbes that are hard to kill by phagocytosis, and it does this by enhancing phagocytosis and antigen presentation. The main polarizing cytokine that causes this is IL-12, mainly occurring when macrophages present antigen. Interferon gamma also triggers this response, and as we know, interferon gamma can come from natural killer cells. The main transcription factor is called TBEP, and this causes more interferon gamma release. Interferon gamma works on macrophages to make them more aggressive. There's more lysosomal enzymes, more secretion of pro-inflammatory cytokines, and it also increases the presence of MHC and co-stimulatory molecules on the macrophages, so antigen presentation is more likely. So as you can tell, macrophages initiate a Th1 response to in turn give them a boost. Hopefully, you can also tell that interferon gamma makes a T helper 1 cell and is produced from them. So this interferon gamma can go to more naive T cells and make more T helper 1 cells, and this is called amplification, because it's like a positive feedback loop to get even more T helper 1 cells. The T helper 2 subtype is in response to helminths, which are worm-like parasites, and it also promotes tissue repair. The main polarizing cytokine is IL-4, and it's usually released by dendritic cells and mast cells. There are 2 other cytokines that lead to the T helper 2 subtype that are released by damaged epithelium, and they're called IL-33 and thymic stromal lymphopoietin, and you'll see why this is particularly needed later. The transcription factor is GATA3, with the resultant cytokines being IL-4 again, so you get amplification, IL-5 and IL-13. IL-4 and IL-13 encourage an alternate macrophage phenotype, making them anti-inflammatory. The main cytokine that should trigger you to think anti-inflammation is IL-10. The main action of Th2 cells is carried out by IL-5. IL-5 sensitizes eosinophils to the parasite. So whenever you think of parasites, think eosinophils and IL-5. IL-13 also enhances mucus secretions and encourages gut peristalsis in the GI tract. In the GI tract, you want low inflammation, hence the Th2 response leading to anti-inflammatory macrophages and encouraging gut peristalsis to move pathogens along is preferred. The T helper 17 subtype, similar to the T helper 1 subtype, greatly encourages inflammation and is usually triggered by bacterial and fungal infections. IL-6 and IL-21 are needed for the Th17 response, which usually comes from the dendritic cell presenting antigen. The transcription factor is called ROR gamma T, and the resultant cytokines are IL-17 and IL-22. IL-17 increases neutrophil recruitment, so more neutrophils are in the infected area. IL-22 acts on epithelia, encouraging it to secrete defensins, which are antimicrobial peptides that neutralize pathogens. The T helper 17 subtype plays a key role in many inflammatory diseases like inflammatory bowel disease and rheumatoid arthritis. The T follicular helper response serves to activate B cells. They have multiple polarizing cytokines, mainly being IL-6, TGF-beta, and IL-23. The transcription factor upregulated as a result is called BCL6. The resultant cytokines they produce is IL-21, IL-4, and interferon gamma to mature and activate B cells and encourage them to produce antibodies specific to that pathogen. Eventually these B cells can become memory cells, so in the event of future infection, the response is much quicker. Finally, we have the T regulatory cells. This T cell subtype is initiated when the infection is cleared. Tissues start producing TGF-beta, which is the main polarizing cytokine for Tregs, and Tregs promote tissue repair. The transcription factor for their response is FOXP3, and the resultant cytokines are IL-10 and TGF-beta. By releasing TGF-beta, Tregs stop other T cell types from being produced and instead create more Treg cells. Again, an example of amplification. IL-10, as we said before, should make you think anti-inflammation. This will facilitate an environment where tissue repair can begin as the infection is cleared. As these responses activate T cells, and these T cells only serve to affect other cells, this type of adaptive immunity is called cell-mediated adaptive immunity. So, to summarize, there are different T helper subtypes. Each have their own polarizing cytokines and transcription factors that induce them to create further cytokines that mainly affect other cells further downstream in the immune response timeline. The Th1 response mainly increases inflammation by making macrophages more aggressive. The Th2 response mainly sensitizes eosinophils to parasites, but they also promote barrier defenses and tissue repair. Th17 responses are highly inflammatory as it enhances neutrophil recruitment, and in chronic inflammatory diseases, a heightened Th17 response is often seen. The T follicular subtype serves to activate B cells, which will then go on to create antibodies more specific to the pathogen and go on further to create memory. The T regulatory cell response occurs after an infection is cleared. They promote tissue repair through the release of anti-inflammatory cytokines. So that's the end of the content section. Hope the video was helpful. If it was, leave a like and please share so everyone can benefit, and subscribe if you want to carry on this journey. And leave a comment if you have any questions or any requests of videos you want to be covered. Thanks very much for watching. See you in the next video.